Hello, everybody. Welcome to today's webinar. We're very excited to have you join us for the conversation today. We're going to cover the integration of three really relevant current areas for drug development: rare diseases, innovative trial design, and model-informed drug development. And you're going to see how tools developed for multiple applications come together to accelerate medical product development for challenging conditions. Let's go to the next slide. Just a few housekeeping notes. Everyone is going to be muted. The chat box is going to be disabled. And if you have questions, please use the Q&A box. And they will be answered either in writing or at the Q&A session at the end. The webinar will be recorded, so mind that, and we will share the recording after that. And with that, we will get moving with my introduction. I'm Klaus Romero, Chief Science Officer for the Critical Path Institute, and I'm just going to give you a very short introduction for how to embrace the learn and confirm paradigm in the context of the topic for today. Let's move on to the next slide. So the bottom line is this. We're going to be talking about the transformation of data into information, but importantly, the transformation of that information into actionable insights to drug development. Hit it one more time. It all hinges on the ability to integrate as many relevant data sources as possible. And CPATH has the ability to do this in a unique way. But then the data integration itself is a component of this, because then all those data sources need to be made ergonomic for analysis. And that is what you see in the second box of curation and management. But then once you have the data speaking the same language, you need to make sure that you're able to build the necessary analysis subsets that you need in order to run the kinds of solutions that you're going to hear today. That's where the first component of the learn and confirm paradigm happens, as you see there with responsive curation. But once you have the subsets, you build the actionable solutions in a collaborative manner through the public-private partnerships that we coordinate. And if you hit it one more time, that's the last component of the learn and confirm paradigm, because with that, we're able to identify what relevant additional data are needed to then be integrated again to have these tools be continuously evolving. And that is an important item for today as a take-home message. These models and these tools, once they're generated, they can't just be thought of as we're done. They need to be constantly evolving, and that was the nature of the underlying work that you're going to hear today. So let's go to the next slide. With that, let me introduce you to Dr. Luke Kosinski, my friend and colleague. He is a true Renaissance man. He is both a philosopher and a natural philosopher as a scientist in his role as senior quantitative medicine scientist at CPATH. He's going to walk you through the first component of integrating the methods together. So Luke, take it away.
Thank you so much, Klaus. So this talk is going to be split into two sections. I'll be doing the first part covering a few select innovative trial design methods, then I'll turn it over to my colleague, Dr. Zihan Shui, to talk about the tools that have been developed for these methods. So next slide, please. Okay, so innovative trial design methods they depart from the gold standard clinical trial. So we think of the gold standard trial, we have a single treatment group, a single control group. There's a lot of features of it, and there's different ways we can kind of adjust it for different reasons. And I'll talk about just a couple select methods today. I'm not really going to go into any of the statistics. This is going to be very high level in terms of the overview. I'll just give you an introduction to a couple of these methods and what they're designed to do. So next slide, please. So the first method I'll talk about is response adaptive randomization. So we think of the typical clinical trial. It tends to use a predefined, maybe arbitrarily defined, chosen allocation ratio between a control and a treated arm, usually a 1-to-1 or 2-to-1 ratio. However, there's reasons why a sponsor may want to try to maximize the number of patients on treatment. For example, there's ethical considerations around keeping a patient on a control medication, especially when we think the treatment might work and we have a severe disease. So one possible solution is to use information to kind of decide the allocation ratio. So in this case, we have subjects being allocated based on the evidence in favor of treatment. And again, I'm not going to get into the statistics of exactly how we know this, but we basically use some prior information to get an expected patient probability of failure on each treatment and use that to inform allocation while preserving power and sample size. So we look at this example I have here. We start with the 1-to-1 allocation ratio. We use some of the prior information to get an expected patient probability of failure, and then we adjust the allocation ratio to have more patients on treatment while still maintaining that power and sample size. Okay, so the next method I'll talk about is platform trials. And again, this is just a very brief introduction. Dr. Colin Hovinga in a later talk will discuss this in more depth. The basic idea here is that with the typical gold standard trial, we have a single treatment compared to a single control arm. Here, with a platform trial, we have multiple treatments compared to a single control arm. And they're usually set up in these multi-arm, multi-stage designs where we have interim analyses that are done. And then if we have any treatments that are found to be pretty hopeless, like if we are able to tell from the interim analysis that there's really no hope of this medication ever achieving a statistically significant difference between groups, that treatment can be ended sooner. We can save on time and all the time and energy and money on continuing it, and then focus on the more promising treatments. And so this is more efficient than the typical gold standard trial because of that, and also because of you're able to test multiple treatments against just a single control arm, and then stop them for futility if need be. Okay, and then the last method I'll talk about today is randomized delayed start. This is, I think, an interesting method. It's very similar to the gold standard trial, except with a bit of a difference. So here we also have a single placebo group, which here is called the delayed start group, and then an early start group. And the difference between the randomized delayed start trial and a gold standard clinical trial is that partway through the trial, the placebo group, the delayed start group, starts treatment. And the idea here is that we want to try to identify a symptomatic drug from one that is actually affecting the disease progression. So the idea is that if both groups are randomized, then each group should have its own baseline, a similar baseline disease progression. And if when we switch the placebo group to treatment, and it's only affecting the symptoms, then it hasn't affected the underlying disease progression. By the end of the trial, both groups should have had about the same amount of disease progression, and they should come back together. Whereas if we have a treatment which is actually slowing the rate of disease progression, then that would happen first for that early start group. And in theory, the two groups should remain differentiated by the end. Now, of course, the statistically verifying this is challenging, but that is the basic idea of how this is supposed to work. Okay, so that's everything I was planning on covering for the methods. I'm going to turn it over now to my colleague, Dr. Zihan Shui. Dr. Shui is a quantitative medicine developer at Critical Path Institute. He has a Bachelor of Science in Math and Applied Math from Beijing Institute of Technology and a PhD in statistics from Florida State University. He has worked closely with data managers to maintain Shiny apps for online platforms and preparing modeling subsets for Parkinson's, Alzheimer's, and polycystic kidney disease. He has also contributed to building multiple GUIs, including GUIs for polycystic kidney disease, type 1 diabetes, and all these three methods that I just talked about. So please take it away, Dr. Shui.
Thank you for the introduction, Luke. Can you hear me?
Yes, we can hear you.
Yeah, great. So this is one of the web apps we developed for RAR. It contains two functions, power calculator and sample size calculator. This slide shows how power calculator works. Users can calculate the statistical power by choosing observed success rate of treatment or control arm, a total number of patients, and significance level. A plot of statistical power by the same settings but different total number of patients is generated as well. For example, given the parameters on the left hand, we can get the statistical power is 0.65, which can be reflected as the red dot in the plot. And this plot also tells us how the statistical power change from different sample sizes. And we also develop a web app based on existing Alzheimer's disease CTS tool by adding a RAR module. Next slide, please. This is one of the web apps we developed for platform trial based on an in-house CTS tool developed for Parkinson's disease. As we can see, this tool can now simulate up to 5 treatment arms. And from the right side, we can see that users can also choose to conduct an interim analysis and provide a milestone at the interim analysis to check for futility. Next slide, please. Here shows a hypothetical scenario aimed at demonstrating the capabilities of the tool. The upper plot shows the simulation comparing a drag arm with control, and the lower plot shows the same at the interim analysis. For this specific example, the futility analysis at the interim shows that the sponsor could continue this arm. And we also develop another web app, which allows users to plan multi-stage trials and evaluate currently running trials for early stopping due to futility at an interim analysis. Next slide, please. Yeah, this is the web app we developed for randomized delay start. Again, the CTS2 for Parkinson's has been modified to allow for randomized delay start. Users may choose whether to run a delay start trial and set the months at which treatment switching occurs. They're also given the option to choose the hypothesis type, slope focused, effect size focused, or both. And next slide, please. The output for the CTS2 shows the trajectory of 3 arms, a hypothetical control arm in red, where no patients switch treatment, the delay start arm in green, and a treatment arm in blue. The output also gives very steady characteristics which reflect the user's inputs. From these outputs, a user can access how well a delay start trial is expected to run. Users can access details about the inputs, statistical power for comparisons, and for individual hypotheses, as well as visually inspect the simulated disease trajectories for the different treatment arms. So these are part of web apps we developed for MIT DBA. Thank you for listening, and I'll give back to Luke.
Thank you, Zihan. So next up, we have Dr. Colin Hovinga, who completed his Bachelor of Science degree and Doctor of Pharmacy from Creighton University in Omaha, Nebraska. He then pursued a residency and fellowship in pediatric pharmacotherapy with emphasis in pediatric neuroscience at the University of Tennessee in Memphis at the La Bonheur Children's Medical Center. Dr. Hovinga had additional training at the Food and Drug Administration's Office of Clinical Pharmacology and has a master's in epidemiology from the University of Tennessee's Health Science Center. Dr. Hovinga is recognized as an expert in pediatric pharmacology and the use of real-world data for drug development. So please take it away, Dr. Hovinga.
Thank you. I'm going to add to some of the comments that were in the last session. Hopefully, we can give you some examples of trials that are ongoing, and it'll hopefully demonstrate some of the concepts that were relayed. Everyone can imagine, in rare diseases and in pediatrics, they often share some of the same concerns. There's one of the most difficult things as a trial investigator is finding patients and identifying individuals that might be eligible for a clinical trial or parents willing to give consent or the individual giving consent. And so one of the limited populations really lead to many sites unable to enroll subjects in a clinical trial, which means also sponsors have to find new sites to enroll patients. In many instances, particularly if there's not good control data, there's concerns of randomizations to placebo, and then the control arms, we have to duplicate them for each study if a randomization of 1-to-1 is used. Trials are lengthy and typically fixed in duration. It's like, and if you turn in your paper and you can't get it evaluated on an ongoing basis, you get the results at the end. So fixed trials, lengthy in duration, and in many instances, it costs a lot of time, energy, and resources to do that. And then what ends up happening if you work in this space is that lessons learned within one clinical trial don't get passed on to future. So the data doesn't inform future research in a particular area. Treatments are done at a time. In cases of what happens in smaller diseases, that's a significant problem, particularly when it's anchored to sample size, right? If there's a disease state for which there's maybe 3 treatments and your population has only a small number of patients that fit the study, it's difficult sometimes to be able to identify those subjects. In real world, we often know that there are, to generalize results, patients aren't just on a single therapy. And it's often unlikely that a single therapy for really complicated rare diseases may be effective. In the case of a platform trial, combination therapies are an option. One of the things I think that to me is often obvious about the platform trials is that one of the most difficult things when you're starting up a study that's timely and takes a lot of resources, it's the infrastructure for a clinical trial is transitory. Trial one may come out, identify sites, go through contract budgeting, IRB, training, etc., only be dismantled after that trial is done. And so you lose that infrastructure that only has to get recreated the next time another study is done. Next slide. Now, master protocols really are sort of the framework that we'll talk about. Some of the nuances that are different from traditional studies is that there you have a standardized protocol. There's a protocol that's filed specifically, has its own IND, which may or may not be a sponsor. It could be an academic institution, it could be a CRO. And then you have the drug arms that are included in there. The studies drugs are classified within appendices. The study protocol typically is a standardized, maybe a menu of endpoints, as well as procedures. The appendices may have specific things that might be required, particularly to a particular drug, such as like pharmacokinetic sampling or a unique safety assessment. But for the most part, the study protocol is done and fixed. And so cell sites and infrastructure can be trained efficiently, and contracts and budgeting relatively streamlined and expedited. Now, multiple therapies can be done concurrently. In many instances, if there's a control group within the trial that's prospective, you can share it within there, as it was alluded to earlier. And the trial design, sample size, randomization can learn and can be adaptive, because this is a unique ecosystem for which the learn and confirm paradigm, can be well executed. Oftentimes, there are predefined interim analyses for these trials, and so you can decide, if there's early termination, for example. And then existing data can go on and inform the statistical plan and model for future studies within this platform trial. Next slide. Now, platform trials can be pretty much any phase. They use, we talked about the modeling in the first, so I won't go into great detail. Platform trials probably take a little bit more upfront planning, because besides your modeling, the monitoring plan, a lot of rules need to be created about decisions, and then data access and who actually owns the data. In many cases, platform trials may be owned by, the trial itself may be owned by an external entity from a sponsor, and the negotiation of, say, like control patient data can be owned or shared with that entity. So all that needs to be looked out. Next slide. Now, I will not go into great details for this, just because they were talked about earlier. Master protocols really fall into three buckets. Basket trial, what I would say, if I was going to give a sales pitch for a platform trial, or basket trial, is it's a drug trying to find a home and using a mechanistic model. And so in this case, what you're doing is you're looking at a single drug targeted against multiple diseases, and it's that drug that is based on a mechanism that may be beneficial in that disease. An umbrella trial is what people often think about as one type of platform trial, and that's where you can study multiple interventions simultaneously with shared control groups. In this case, it's one disease, multiple treatments. And the last one I think we've spent a lot of time talking about is the multi-arm, multi-stage adaptive trial, or MAM study, in which you have the ability to make decisions as the trial is unfolding in various points of the study. Next slide. Now, the adaptive requires multiple stages. You have a baseline model. What I really want to get to as the last point is that typically it has an allocation rule that decides the imbalances in arms that can be justified based on your looks. There's sampling. You can change the sample size. It can decrease, increase based on the interim analysis. And then stopping rules, that if there's a futility signal that's registered between, in a study, and whether efficacy is established or safety or toxicity is a concern. And so you don't have to wait to the end. The next slide. Now, just some quick examples. The Intuitive study is an example of a basket trial where basically three different, NF is the disease subtypes there are listed. There are various different schwannomas listed, but one drug is studied for each of those indications, each of those subtypes. And then what happens is, after a set number of patients enrolled, and there's absence or of disease progression or progression, those decide whether or not you go on with the study. But again, these are just subtypes of a single disease entity, one drug against them. Next slide. Now, Healy platform trials at ALS, this uses a primary endpoint as the ALS-FRS, and what you're going to see here is that there are up to 4 arms going simultaneously with a 3-to-1 randomization, so 3-to-1 for each treatment for placebo, and then the trials continues on. What happens is, as one study arm completes, you can add additional study arms in an ongoing basis with a shared control group. Next slide. And the last one I want to talk about, it's not a rare disease, but it's an adaptive design, and it's one of the most well-known one is I-SPY. I-SPY uses basically subtypes of breast cancer, neoadjuvant breast cancer, a treatment. And what it does is it basically looks at HER2 status, and what it can do is an adaptive design, like a MAM study, where in this example, if you look at drug A, the outcome of drug A, you're going to see that the drug A benefited those patients versus those in drug that are in the green and blue. Those arms are going to be pulled out of the study. And the example for drug B, no one benefited for the trial can stop early. And the same thing, you can match and do the controls to mirror that response. Next slide. Now, one of the things that's always, I think, gets hidden in these studies, and this is something I think that's most challenging to talk about when you're dealing with this, is the patient perspective. So, like, one of the things that's nice about platform trials is that you get access to more treatment arms at a single site, right? That same infrastructure, you may have multiple choices. You can get more efficient completion of studies. You don't have the ramp up phase as much that you need. The site's well trained, and et cetera. They generally, persons with lived experience generally feel data sharing is positive. So they feel like they're giving to the good because their data may be used to inform future trial designs. What may be different and confusing to people is oftentimes there's a two-stage consenting process, because unlike traditional trials, is you have to consent to enroll in the protocol, but also consent to be randomized to a specific treatment arm. So, first step is getting you in the platform trial, for example, or master protocol. Then it's the risk benefits within a specific arm. Blinding can be a concern, and we can address that in the Q&A. But I want to put those things out there. One of the biggest concern usually from individuals is, I don't know how many individuals on this call have enrolled patients in clinical trials, but is the loss of autonomy that they might fear. Because in this process, you're agreeing to be randomized to the platform trial. And so you don't get to choose which treatment arm. You have to be randomized to the ones that are available, whether or not, so some may be less interesting to families as they participate in that. Generally, what you're going to find is that, the other balancing things like loss of this loss and concern is that, the sharing of data and the more efficient access to treatment, those oftentimes are usually felt as sort of the trade-off that people feel valuable and whatnot, and so that they are more eager to participate in those clinical trials. And I think that wraps my presentation, and off to the next.
Thank you so much, Dr. Helvinga, for that great talk. So next up, we have Dr. Angela Genge, who is recognized internationally for her work in clinical trial design and execution in the field of rare disease, particularly ALS, myasthenia gravis, and FSHD. She is the executive director of the ALS Center of Excellence at the Montreal Neurological Institute at McGill University, where she recently stepped down from executive director of their clinical trial unit after a 20-year tenure. She serves as scientific advisor for a number of clinical trial networks, including ENCALS, CALS, and she is a member of the executive committee of the WFN ALS section. Her current activities include CMO of Curalis, global PI of ALS Pharma, and she serves as an advisor for ALS, FTD, myotonic dystrophy, FSHD, and myasthenia gravis, among many others. She's a regularly invited speaker on many topics, from novel therapies in neuroscience to regulatory challenges internationally, and she has received a number of awards, including the Forbes Norris Award, the Wings Over Wall Street Award, the YWCA Woman of Distinction, and Founder Award from Community Cares. She advocates tirelessly for therapeutic innovation in neuroscience and access to therapeutics for the rare disease community. So please take it away, Dr. Genge.
Thank you, Luke. Next slide, Luke. So what I'm really introducing you to is the concept that is really an extension of what we've had so far. We've had a very good review, an excellent review, in fact, of the role of platform trials and adaptive trial designs, and some tools that now exist for these approaches to trial design in rare disease. The adaptive trial designs that have been initiated in diseases that, in fact, have no particularly great therapeutics is important for the patient and the clinical trialist community, because the need for speed exists. And I really use ALS as a prime example. Amyotrophic lateral sclerosis, as you know, is a rapidly progressive fatal disease in which patients go from perfectly healthy to demise within 2 years, 2 to 5 years, but some as quickly as 9 months. So there's a tremendous interest in changing ALS from a fatal to a treatable chronic condition. And the adaptive designs have entered into the field quite quickly over the last few years, with the platform trial, with adaptive designs being used in individual development programs to shorten the time to application for registration. The adaptive design that is, other than the Healy platform trial, which Colin just mentioned, the adaptive design that I am seeing most frequently is several different versions of a seamless 1-2 or a seamless 2-3 design, with a heavy emphasis on open label extensions being built into programs to really enable additional safety, tolerability, and efficacy data to be ready for your registration package without having to repeat your pivotal trial. This morning, I'm really going to comment on the front end of this process, not the back end. Next slide, please. With the FDA launching a new pilot program, it's actually encouraging a number of small biotechs to get in front of the FDA for advice on their trial design. But there's one area that is sometimes overlooked as a place for efficiency in drug development and clinical trials, and that is the phase one period, that first in human period in drug development. Next slide, please. Every developer of a new investigational product is very well aware of the IND enabling studies that are required prior to entering a patient or a person or participant. However, the really important step and role that phase one first in human studies play, I think, cannot be overemphasized. And to do this in an appropriate manner actually is a point of conversation with the patient ambassadors in some of the rare diseases. When we look at phase one trials, which we all know are for safety and tolerability as its primary or co-primary reason for existing, there are essentially two main approaches. One is to take the investigational product into healthy volunteers, and the other is to take it directly into a patient population you hope to develop the drug for. And in the case of the healthy volunteer studies, these are actually very well developed, well understood methods of entering into the clinic and preparing you for your multi-dose and your proof of concept phases of your drug development. It is not always obvious whether you should do a phase one program in healthy volunteers, or whether you should go straight to the patients you wish to target in your development program. So I simply want to review some of the main pros for each of these approaches, and what has led, in fact, to different approaches in ALS. both approaches are used in amyotrophic lateral sclerosis for the reasons that I'm about to outline. So in phase 1 clinical trials using healthy volunteers, the overwhelming rationale that clinical development professionals bring to the table in deciding to take the healthy volunteer approach is the speed with which you can do your single ascending dose, reach your maximum tolerated dose in your phase 1 study. It actually is most conducive to small molecules with an oral administration, in my experience. And the healthy volunteer studies play a particularly important role if the pharmacokinetics or your PK modeling in the human circumstance, which is who we're treating, has any reason to be different than in your non-human primates or in your IND enabling studies. So what really happens in those studies is that you get very intense PK modeling, frequent blood draws, and it becomes extremely important in the entire development package that you fully understand your PK profile in human participants. The other the next main rationale for going with a healthy volunteer study is really if you're going to be doing frequent dose escalations and you expect to need a fair number of cohorts as you get to your maximum tolerated dose. This is actually far more difficult if you're recruiting from a rare disease population. So I think that is fairly obvious for people. Less obvious, I think, is the issue of the safety. Potential side effects play in two different ways into the decision of going into healthy volunteer or straight to patient phase 1 studies. The potential side effects of a novel therapeutic are important in assessing the risk benefit in a healthy volunteer as compared to a patient population. And so when you're looking at a patient population which is rapidly progressive and fatal, which is ALS, you're looking at a very different, even though a rare population, than conditions such as facioscapulohumeral dystrophy, which is the most common form of adult muscular dystrophy, but again is a very slowly progressive disease condition without systemic complications of the underlying disease. So when you look at a healthy volunteer study, you want to be able to use a healthy volunteer study in situations in which the underlying condition may have its own complications, myotonic dystrophy being an example, that would affect your assessment of the adverse events that you are seeing in your phase 1 trial. I think the final, probably the most frequent reason people will do phase 1 healthy volunteer studies in the rare space is in fact the frequency of the patient population, the incidence and prevalence. So again, it's back to, in fact, efficiency, but using a healthy volunteer population allows you to get to your proof of concept phase of your trial with a relatively efficiently, whereas if you are developing a therapeutic for Fragile X syndrome, the time it will take is such that it becomes a very inefficient phase 1 program. Next slide, please. So why would you make the opposite decision? Why would you make the decision to go straight to patients in a phase 1 study? I think the most direct reason that we're currently using to justify our novel therapeutics in ALS is the type of intervention required for drug delivery. We're currently in a phase where intrathecal delivery is now being used in amyotrophic lateral sclerosis for antisense oligonucleotide therapy, gene therapy, siRNA therapy. And in all these instances, doing a healthy volunteer study makes less sense ultimately when you're looking at how you're delivering your investigational product. The one-time dosing approach also is a second reason why people are really moving toward phase 1 in the population you want to study, particularly with gene therapy. The use of patients in phase 1 studies in which you have a relatively healthy patients, if I may say. ALS, FSHD are both conditions in which the disease actually is very limited to either the motor neuron population or the muscular dystrophy, without any systemic complications of these conditions inherently. So these patients and the patient ambassadors are often very keen to have the drug go straight to the patient population. The final main reason for a patient-based phase 1 study is really if there are safety concerns that are specific to the target condition, and inherent in the future planning is biomarker information that you can use as you move from phase 1 to phase 2. Next slide. So the recommendations really are as follows. Look at your investigational product. Look at the phase 1 as part of your overall clinical development plan, giving you information not only on your product, but also on the biomarkers that are under development and the patient population and engagement. Look exactly at what else you will find in your phase 1 study that may inform your decisions on your trial design, your ability to use an adaptive design. The balance is between information and speed, and ultimately the patient advisory councils, particularly in some of these diseases, are quite knowledgeable and can give you some very specific guidelines as to which direction they feel you should take. Next slide, please. Thank you very much.
Thank you so much, Dr. Genge. That was a very informative talk. So last but not least, we have Dr. Hao Zhu. Dr. Zhu is the director of the Division of Pharmacometrics at the Office of Clinical Pharmacology, Office of Translational Science, Center of Drug Evaluation Research at the U.S. Food and Drug Administration. Dr. Zhu received his PhD in pharmaceutical sciences and master in statistics from the University of Florida. He started his career in modeling and simulation teams in Johnson & Johnson and Bristol Myers Squibb. He joined FDA as a pharmacometrics reviewer more than 16 years ago. Dr. Zhu has been a clinical pharmacology team leader for more than 6 years and a QT-IRT scientific lead for 2 years. He became the deputy director at the Division of Pharmacometrics, and his division reviews the pharmacometrics-related submissions and supports pharmacometrics-related policy development. So please take it away, Dr. Zhu.
Sure, thanks, Luke, and thanks a lot. I think we had a very nice presentation from other presenters. So I would like to continue that discussion by sharing our experience and understanding of using modeling as a tool to facilitate the clinical trial design and improve clinical drug development. So before getting into the details, I do have a disclaimer. So what I'm presenting here today are just my, are just my personal views and should not be considered as an FDA's official position. Again, my presentation includes the following 3 sections. I'm going to have a brief introduction about the concept of MIDD as a technical tool, as well as a regulatory tool. Then I'm going to share some case examples based on our review experience. Then I'm going to conclude with some take-home messages. Next one, please. So let's begin by looking at what we mean for MIDD. So MIDD, or we call model-informed drug development, actually has 2 meanings if you want to look at it. So from one side, it is a technical concept, which means that we're trying to apply a broad range of quantitative models to facilitate drug development and decision-making. On the other side, MIDD can be considered as a regulatory tool, which is designed to promote earlier interaction between drug developers and tool developers with the agency. So let's begin by looking at what do we mean as MIDD for regulatory tool. So again, this is really from narrow scope. We're really talking about MIDD parent meeting program. We know this program is jointly administered by CDER and CBER. The Office of Clinical Pharmacology is the point of contact. And of course, any sponsor who is a drug or biologics developer with a product registered as IND, NDA, a BLA could be a potential user of this program. And the intent of the FDA to create this program is to generate an environment that is designed to increase stakeholder acceptance of MIDD approaches. It is used to develop standards and best practices that leads to consistent application and evaluation. And it also is intended to increase the capability and also expertise to address the growth demands and innovation. And you can see the figure to the left bottom side. Actually, there is actually a publication from the IQ. It actually shares the industry's experience by using this MIDD program. I want to also mention there are many successful cases in rare disease areas or neurological disease areas where we try to discuss with the sponsor on their specific drug development program in order to target some of those challenging diseases. Next one, please. Now I'm going to switch gear to talk about MIDD as a technical tool. So essentially, MIDD in this domain really talks about how we can develop and apply exposure-based biological and pharmacological models derived based on preclinical and clinical data sources to address drug development or regulatory issues. If you really open a toolbox of the MIDD, you will see there are a lot of modeling approaches available. For example, we can try to understand the concentration change over time by using PK modeling, which generally includes population PK modeling or physiologically PK modeling. And also we can try to link the concentration changes to the subsequent lab value changes or pharmacodynamic value changes, and essentially link to the clinical outcome changes. That is what we call exposure response modeling. And of course, we can also try to use mathematical equations to characterize the underlying disease progression in general population, or try to understand what the patient features may potentially affect the disease progression over time. That is what we call disease modeling. So we can use all the modelings in combination to assess clinical trial design. Not to mention, in recent years, we have more complicated modeling structure where we try to link the underlying biological process into different layers. For example, tissue layers, organ layers, whole body layers. And this is what we call a quantitative system pharmacology modeling. And of course, we use some highly empirical modeling as a new addition to the toolbox of MIDD, which includes artificial intelligence, machine learning models. We try to understand, can we actually include real-world data, real evidence as an alternative source of information. Also, we try to incorporate the digital biomarker information. So all these tools we can use in isolation or in combination, but the goal is the same. We try to really modernize new drug development, and so that we can improve the patient care. Next one, please. And now I'm going to really focus on how MIDD can improve clinical trial design. We have the very fabulous discussions earlier showing how we can do things differently with the innovative trial designs. Now we can actually focus on some of the aspects. For example, we can use MIDD to select what is appropriate clinical development approach, how we can inform patient selection, assess the patient randomization, optimize dosing, choose sampling frequencies, sampling points, and identify primary, secondary endpoint, and conduct relevant analysis. Next one, please. So now I'm going to share one example. This is really in a rare disease for neurological disease domain, where we try to justify why neurofilament light chain can be considered as a reasonably likely surrogate endpoint for accelerated approval for drugs for treating SOD1 ALS, which is a very rare type of ALS patient. So essentially, we look at all the pieces of information from different angles. Number one, we look at what is the biological plausibility for the NFL change that can be linked to the subsequent clinical outcome changes. Also, we try to understand, do we get information on the prognostic values of the NFL? Also, we try to establish the relationship between the tofersen-driven NFL reduction and clinical functional change. So you can see the figure actually to right-hand side, there are two figures on top that shows is the prognostic value of NFL by looking at the historical data. Also, the clinical trial data, which are actually showing by the two figures in the bottom, are actually support there is a correlation between the tofersen-driven NFL changes and clinical outcome changes. All the pieces of information put in together actually help us to determine that NFL can be considered as a reasonable likely surrogate endpoint, which can be applied to support accelerated approval for drugs that actually can be used to treat SOD1 ALS. Next one, please. And the second example is not in the neurological space, but it actually can be applied to probably in rare disease or in neurological space in the future, where we try to link efficacy and safety signals all together. And the drug we're trying to talk about is a bispecific antibody. We understand bispecific antibodies can trigger very severe adverse event that is called cytokine release syndrome, which can be sometimes life-threatening. So in this case, we're trying to use population PK model to link dose to the concentration changes. Then we try to generate the QSP modeling that can link the exposure changes to the IL-6 level changes. And subsequently, the changes in IL-6 level can be linked to the incidence of cytokine release syndromes. And in this setting, we can actually understand, given this dose level, what is the concentration change, and then what is the potential for this patient to develop a CRS. In the meantime, we can also try to understand, can we actually get a reasonable suppression on the tumor growth? So this actually gives us a reasonable balance on what the dose we're trying to achieve in order to control the tumor growth. In the meantime, we can actually manage the incidence for the very severe adverse event, which is a CRS. And next one, please. And of course, I'm going to talk about, there's always, we can use modeling simulation to justify, can we actually extrapolate efficacy findings from one population to another population. Essentially, this is a disease model we try to establish in patients with schizophrenia. You can see, the outcome changes fairly similar between pediatric patients and adults, even with or without a drug. This actually becomes the basis for the FDA to actually establish a new policy allowing the full extrapolation of efficacy findings from adults into pediatric patients. Next one, please. And the brexpiprazole is the first drug actually applied to this policy, and you can see the usage of brexpiprazole has been extended from adults to pediatric patients 13 years and older for treating schizophrenia. And next one, please. Last but not least, I'm going to conclude my presentation. I want to emphasize that MIDD is really a powerful tool to support new drug development and regulatory decision-making, and the scope of MIDD applications has been expanded. So we believe with a joint effort from academia, industry, and regulatory agencies, we expect that the MIDD approaches can be further used to facilitate new drug development and to improve patient care. And next one, please. And of course, I would like to acknowledge Dr. Xiongmei Huang, Dr. Isam Zene from our office, of course, our division members and office members. And of course, I wanted to thank you for your attention.
Thank you so much, Hao. That's a great tour de force. May I ask all the presenters to go on camera to start the Q&A session? Okay, so we have several questions from the audience. I'm going to start with a question that we received about the notion of in ultra rare diseases that may have equivalent biomarkers or equivalent non-biomarker outcome measures. Can a platform trial help optimize sample size? And Colin, I'll get started with you.
Yeah, I think the short answer to that is if the similarity is strong enough, yes, it would be adequate. I mean, I think what you're describing, actually may be either a multi-arm, multi-adaptive study, but also I think that it might get overlooked. It might be eligible for something like a basket trial, if particularly if you have a sponsor wanting to approach multiple disease states at once. That might be an option to be able to do one drug. The one caution I would put as you think through platform trials is that there's situations where if you're dealing with diseases where there are specific genotypes and there's a therapy targeted to those genotypes, generally in platform trials, most people would be hesitant to include those molecules because randomizing someone with a genotype that has no potential for benefit, for example, would not be something where someone would feel good about enrolling or ethics review.
Thanks, Colin. And you started a great segue into the second question from the audience. And I'll pass it on to Angela to build on it. The question is, are basket trials feasible and a good option for diseases with high heterogeneity and differing genotype phenotype, or would one utilize this type of design for the same disease with slightly different genome and phenotype? Angela, do you want to build on this?
Happy to. I appreciate Colin's insights. So I look at basket trials from really building on what was already said as a method relatively early in drug development to look at the mechanistic component of the investigational product. And I think in those instances, basket trials can actually be extremely useful, especially if you have a biomarker that is ready for clinical validation and is directly linked to the investigational product. We've recently had one in ALS and FTD that targeted TDP43, a component of TDP43 pathology. And what it really does for development is it gives you all the safety and tolerability, because safety and tolerability is the same across, humans are humans. And with FTD and ALS, there's no particular systemic difference in those two diseases that would complicate your interpretation. So I think it's actually basket trials early on, especially with our innovative therapy that are targeting pathways as opposed to that are shared across a number of different diseases. I think basket trials actually are one of the better approaches early on, and then you choose your target disease for your registrational package for your pivotal trial.
Excellent. Thanks, Angela. And so that is, again, a great segue. The audience was really tuned in. Another question from the audience regarding the primary way to analyze the data for a potentially disease-modifying treatment. So, Luke, do you want to take this one? The question is essentially as follows. If you have a disease-modifying drug, could you and should you base the primary statistical analysis based on the magnitude of change of the rate of progression versus just the net difference between arms at the end of the follow-up period?
That's a really good question. Statistically, you definitely want to look at the difference in arms at the end of the follow-up period. But when you're trying to evaluate whether there's really a disease-modifying effect, one approach you can take is exactly the one that was mentioned. There's a set of hypotheses was developed by Atul Bhattaram and a few other authors for looking at slope-based approach to try to figure out whether there really is a disease-modifying effect. And then there's other approaches that have been proposed. But yeah, so yes to both. Actually, you want to do both.
Perfect. Thank you so much. And Hao, I'll pass this one to you. This is a question about those conditions in which the placebo effect is very patent and important to understand. So the question is if you can and should model the placebo effect to better understand how to optimize shared controls in platform trials.
Yeah, I think this is a good question. My short answer is definitely I see the value. You really need to understand, what is the general disease progression, and also on top of that, if there's any additional changes, if you give the placebo to a patient, depends on the disease we're talking about. A lot of times, the observations in the placebo arm is more or less more reflected the general disease progression. So understanding the time course of the changes in the placebo arm is going to definitely beneficial to determine how long is your treatment duration, what is the sample size you're really targeting, and what is the optimal sampling schedules to pick up the appropriate observations in the interim. Even it's any type of, it's probably beneficial for any type of clinical trial design, I would say.
Excellent, Hao. Thank you so much, and thanks to the audience for the questions. So at this time, we have 3 minutes left. I want to thank every single one of you for taking the time today of sharing the relevant information that was shared. I'd like to thank the audience for being tuned in and asking the questions, making this really interactive and informative. You know where to find us. So if you have any other comments or questions or want to get involved in our public-private partnerships, from wherever you are in industry, academia, regulatory agencies, patient advocacy groups, people with lived experiences, please reach out, and we would love to follow up with you. So with that, I think we can move to adjourn today's webinar. Thank you so much, and have a great weekend.